Hello, thank you so much for attending the ORCA Data Jam webinar. This will give you a nice overview of our project as well as some inspiration, some guidelines, and we'll also practice with some research questions and data analysis. Before we get into Data Jam, I just want to talk a little bit about ORCA. So ORCA is dedicated to the protection and restoration of aquatic ecosystems and the species they sustain. We do that through the development of innovative technologies and through science-based conservation action. So these are kind of our main pillars at ORCA, innovative technology. So you may have seen these machines if you spent some time out on the Indian River Lagoon. Those are our Kilroy devices. And our Kilroys take in water 24/7 and analyze it for many different analytes that tell us about the health of the Indian River Lagoon, things like dissolved oxygen, temperature, chlorophyll, FDOM, nutrients. Basically, like you go to the doctor and they might look at your eyes, they might test your heart, they might look in your nose, basically to make sure that you're healthy. We're doing the same kind of thing, but for the Indian River Lagoon. Hopefully, if anything is not healthy, we can try to inform the community and work to change that. We are actually the largest real-time water quality monitoring network in all of Florida, thanks to our Kilroy network. We do quite a bit of applied science research. A lot of our research is focused on ecotoxicity and pollution. We make pollution maps so that we can identify hotspots of pollution in the Indian River Lagoon and really know where to concentrate our conservation and research efforts. Those are kind of similar to weather maps. A really polluted area would be red, and then really good would be blue, with everything else kind of in the middle, similar to a weather map. We have a robust citizen science program, so we have 4 different citizen science projects. I'm going to talk about all those in just a minute. But citizen science is pretty much what it sounds like, teaching everyday people how to take real, reliable data. And then, of course, we have education. So Data Jam is one of our large education projects. We also go into schools and teach lessons and do a wide variety of community outreach events. So we're focused on preserving the Indian River Lagoon. The Indian River Lagoon is an estuary, so it's a nursery ground for lots of creatures, and it's a mix between fresh and saltwater, making it a brackish ecosystem. So 40% of Florida's east coast is bordered by the Indian River Lagoon. It's a pretty significant chunk of our east coast, all the way from Volusia, New Smyrna Beach, down to Jupiter Inlet at the tip of Palm Beach County. This is actually the most biodiverse estuary in all of North America. There's over 4,000 species out here. Really amazing that we have that right here in our backyard to study and to conserve. About 75% of the fish that humans eat will grow up in estuaries. So these are very vital ecosystems for all the creatures that are moving out into the ocean later in their lives. So these are our 4 citizen science projects. We have pollution mapping citizen science. Pollution mapping is where we look at a wide variety of pollutants, both in the water column as well as the sediments. Things like glyphosate, which is a common ingredient in herbicides. Things like nutrients, which can cause excess algae growth and eutrophication. Things like sucralose and acetaminophen, those are human tracers. So lots of interesting information coming out of that project, and you could definitely look at our nutrient data if you're interested in looking at that project for Data Jam. A Day in the Life is our kind of educational-based citizen science program. We have students from 4th grade all the way up to college that all go out on one day in October to take data on the Indian River Lagoon. So they're taking water quality data, biological data, some physical data. So we're kind of getting a snapshot of the whole lagoon on that one day. If you're interested in this data set, you can look at glyphosate concentrations across different A Day in the Life sites. We have our Land to Sea Citizen Science Project. So that is our habitat restoration-based project. That is where we make living and buffered shorelines. So living shorelines are water-based. Those include things like oyster reef balls, oyster mats, oyster bags, like you see in the photo. Basically something that you put out in the water that helps to stabilize the shoreline, reducing erosion, increasing habitat for lots of invertebrates, recruiting new oysters, which help to filter the water, keeping it nice and clean. So many benefits of those living shorelines. And then the buffered shorelines have many of those same benefits. So those are land-based. That involves planting native plants on the shore, especially close to the water, getting rid of things like green grass that need a lot of fertilizer. And those native plants, lots of the same benefits. So reducing erosion, uptaking nutrients that we don't want in the lagoon, as well as increasing habitat. Then we have our One Health Fish monitoring. That's our last citizen science project. So that is where we look at toxins and toxicants in fish from the Indian River Lagoon, specifically fish that people eat. So we've done angler surveys to identify the species that people do eat because we're interested in seeing how those toxins transfer to humans. So things like glyphosate, microplastics, heavy metals, microcystin, PFAS, which are forever chemicals, all those things we look for in our One Health Fish monitoring. And if you're interested in that data set, we actually have a couple different ones to choose from for Data Jam. You can either look at the microplastics in the fish or the mercury. We have a quick little Data Jam video to kind of introduce you to the program, give you some inspiration.
The Data Jam is a competition where students or adults take actual science research data sets collected by ORCA citizen scientists. They get to analyze it, interpret it. Instead of creating some kind of science research paper, they get to be creative and unique, and express and tell the story of that data through some kind of artistic means, whether that's a sculpture, a play, poem, comic strip, an animation, or more. The Data Jam is a chance for students to really express and build on their passion for something. Allow students to explore that passion, throw their data into it, figure out how you can be a passionate communicator, but still convey a scientific message.
So you can see Data Jam is our program where we combine art and science. And this is our CEO, Dr. Edie Witter, talking about how important art has been in her career. She's a deep sea biologist. She spent her career studying bioluminescence, and art has been very important for her and for science.
Let's talk about why art matters. Science is the most important tool we have to explain our world and our place in it. Effectively communicating science is critical to solving some of the world's most pressing issues. Most of the time, science is explained using charts and graphs, but sometimes art can have a far greater impact than simply providing numbers on a graph. I'm a terrible artist. A lot of my career has involved exploring deep parts of the ocean and seeing bioluminescence in ways that nobody's ever seen it before. And for a long time, there were no cameras that could actually record it. It was really dependent on artists to convey the kinds of things that deep sea biologists were seeing. And alter perspectives and inspire new insights, which is why at ORCA we seek to foster the link between art and science with programs like ORCA's Data Jam, a competition that challenges artists to use real science data sets collected by ORCA citizen scientists and come up with artistic ways to share their findings. So please go to teamorca.org today to find out how you can participate and support ORCA's efforts on behalf of the Indian River Lagoon.
Alrighty, so what exactly is the Data Jam? You can see from those videos that it's a competition that merges the art and science. We ask the participant or the team to analyze a scientific data set and illustrate the trends observed through the art. So for this competition, imagination is really the only limiting factor. You can do any type of art you can think of: paintings, sculptures, songs, plays, dance, comic books, animations, film, poetry. We've had people do crochet. We've had people make board games, video games. The opportunities are endless. So whatever interests you, try to build on that. So 75% of your score is going to be the creative portion. That's the majority of your score. 25% is going to be the supporting project description. So that's going to be the written portion. You can find the written portion in the Data Jam guidebook, which you can access from our website, and I'll show you how to do that in just a few minutes. So before we get into that, I just wanted to show a few examples of what Edie said, how art can really alter perspectives, sometimes even more than just showing data. So this is a photo that I love to use as an example. This is actually someone I've been following on Instagram for over a decade now. This is Zach Noyle. This photograph became popularized when it was published in the Huffington Post. And as you can see, this shows a surfer literally immersed in a trash wave. So maybe you could count up that trash. You could tell people the different types of trash. You could weigh it. But is that really going to be as impactful as showing this photograph of someone trying to be in the ocean that is just in a trash wave? Probably not. And this actually sparked action. So once this did become popularized by the Huffington Post, this area started doing regular beach cleanups, and it's actually improved a lot in the years since this photograph was taken. This is another nice example. This is a painting that shows the mutualism between clownfish and sea anemones. And if you look really closely, there's actually data hidden in this painting. So these are all data points. You can see that there's a negative slope occurring from these data points. And this is actually about how climate change has affected the population of anemones and clownfish. So she kind of incorporated the beauty of these species with data to really make an impact. Comic books are always a good option. So one of the data jams that we took inspiration from was the Hudson River Data Jam. The Hudson River Data Jam had this as a nice example, this comic strip. You could obviously do this about the Indian River Lagoon. It would be different subject matter, but again, using art to get a message across about the data. This is a nice game that some elementary schoolers actually won with. They looked at the microplastics. So it's an interactive game. You're a diver, and you can actually dive through the ocean and click on these different fish species. These all represent samples that have actually been donated to ORCA. So this group was interested in fiber microplastics. So you can actually click on a fish. It'll tell you that this fish had one fiber microplastic, the pigfish. Sheep's head had one fiber microplastic. Mangrove snapper had 0. And you can go through and actually look at all these fish that were donated in this interactive game. So, some guidelines for Data Jam. It can be done either individually or in small teams. So teams can be up to 8 people, and we do have different age categories. So you're only going to be judged on your age category, which would be upper elementary, middle, high, or adult. So for upper elementary, 4th and 5th grade is good. If there's a really motivated 3rd grader, they could maybe do it as well. And again, either individually or up to 8 people on a team. You're going to choose an ORCA dataset. You can access those on our website. Again, I'm going to show you how to navigate there. You're going to come up with a research question, and you're going to analyze that data. That can sound a little intimidating, but we are definitely here to help. If you do need any tutorials with Excel or data analysis, anything like that, feel free to reach out to us. We can set up a Zoom. We can have you come to the Center for Citizen Science and get some help. If you're a teacher and you want me to come to your class for a day or a few hours to tutor, we could definitely do that. And then you're going to create. So the fun part, you're going to make any type of art project you can think of. Again, imagination is the only limiting factor. You're going to write your project description. That is from the ORCA guidebook. We give you a template for that. And everything is going to be due on April 25th. We're also going to have a Data Jam showcase. The Data Jam showcase is going to occur on May 8th, But we do want the projects a couple weeks before that, so projects will be due either digitally or you can drop them in person to our Center for Citizen Science by April 25th. Alrighty, so I've been mentioning our website several times and how to navigate to some different resources that you'll need. So you'll visit teamorca.org. You can go over to the tab that says Citizen Science and Education. A little drop down will come down. You'll click School Programs, and then you'll click Data Jam. When you click Data Jam, you'll be brought up to a page that looks like this. So there's a few different tabs: Data Jam Resources, datasets, old winners. So you should definitely explore all of this, especially the resources page. I've mentioned the guidebook several times. That guidebook has all the rules you need, all the guidelines, as well as that written template for the written portion. We have a planning schedule on there. So that's just a suggested schedule of when you should have certain parts of the project done by. We have our judging rubric. So if you want to know exactly what we're looking for judging, you can pull up that rubric and tailor your project to make sure it includes everything the rubric asks for. And then, of course, some inspiration. You can look at past ORCA Data Jam projects as well as some Hudson Data Jam projects. You'll click on this tab to see the different data sets. So there'll be a data set for each project, as well as a short introductory page that will explain the data set, some different variables, have some examples of some different research questions. So definitely check those out as well. And it'll also have the links to our different projects on our website, so you can kind of explore those projects a little bit more before deciding which one you want to do. All right, so these are the different data sets you can choose from for Data Jam. For pollution mapping, you can look at nutrients in the water and in the sediment. So water column or poor water, we call the water that's lodged between the sediments the poor water. So you can compare that across different seasons, different years, water column versus sediment, all kinds of different comparisons. For a day in the life, you can look at the glyphosate concentration in the water, again, across many different sites, many different years. For One Health fish monitoring, you can look at the microplastics, so the different types of microplastics, how many are in different species, total microplastics in different parts of the lagoon, or you can look at mercury. So mercury concentration, same kind of thing across different species, different years. So we're going to practice coming up with some research questions. And then for my high schoolers and my adults, this one's a little bit more advanced, but it's new this year. It's our Sebastian River Ecotoxicity Project. This technically isn't one of our citizen science projects. It's not long and ongoing. It was a one-time thing, but we did use a lot of citizen scientists, and it's really interesting data that is coming out of it. So we figured we would add it to Data Jam this year for high schoolers and for adults. So if you want to look at this data set, you would be looking at heavy metals in muck. Okay, so we're going to practice doing a little bit of data analysis and coming up with some research questions. So the first data set we're going to look at is our One Health fish monitoring data set. As a review, this investigates the pollutants entering into the Indian River Lagoon and how those pollutants might get into the food chain, specifically into fish, and specifically into fish that humans eat. So this diagram kind of shows how a pollutant will enter a food chain. It'll start off with a very small animal that'll consume the pollutant. That will then be eaten by something bigger, and they'll eat several of those small species. So all the concentration that was in each one of those will now be in the consumer. And that'll just keep going up as you go up the food chain. So now you have that secondary consumer that's eating the primary consumer and uptaking all the pollutants that that guy had, and he'll be eating several of them. So those toxicants and toxins tend to increase as we move up the food chain, and we really want to explore that with our data. So what specifically are we looking for? Heavy metals, things like mercury, cadmium, lead, microplastics, different types, Forever chemicals, we call that PFAS, polyfluoroalkyl substances. Those are basically chemicals that never break down. Microcystin, that's a biological toxin, so blue-green algae. And then glyphosate, which I've already mentioned, that's a common ingredient found in many herbicides like Roundup. And we're specifically going to look at the microplastics. So this is how microplastics enter the food chain. Lots of times they start out from large plastics, things like plastic bottles. Over time, those will break down from the salt water, the sun, and turn into microplastics, which will then be eaten by fish and then consumed by humans. Some plastics will start out as microplastics, like face beads and face washes, but a lot of them, probably most of them, are from breaking down. Alrighty, so these are the different types of microplastics we look for. We have fragments, which are hard, jagged pieces of plastic. Those come from bottles, things like hard, sturdy plastics. We have fiber. So that is a thin or fibrous straight plastic, things like fishing line or nets. Pellets, which are hard, rounded particles. Those could be from resin pellets, facial cleansers, which I mentioned. We look at film, so that would be a thin, flimsy plastic, like from a plastic bag or a wrapper, or foam, so lightweight, sponge-like from styrofoam, cushioning, things like that. So we do categorize these different microplastics when we process our fish that are donated. So this would be an example of how the data would look when you pull it up. So this is just a little snippet of the data. The data sets on our website are much, much longer, but this is just a little snippet to help you practice. So we're going to practice coming up with some research questions. Here are a few that I thought of with this data set. I really like this first one. It's nice and easy. Which type of microplastic is the most commonly found in fish from the Indian River Lagoon? So I want you to take a second and think about how you would solve that question. If you want to pause this webinar to think about that for a minute, go ahead and do that now. So how you would solve which type of microplastic is most commonly found in fish from the Indian River Lagoon. For this one, you would just be adding up the columns of the different types of microplastic. So you would add up all the granules, which in this data set you can see is 0. You would add up all the fiber, which you can tell without even adding it that fiber is going to be the most common. Add up all the fragment, all the film, all the other, and compare those. Here's another question we could do. Is there a difference in total number of microplastics and fish species? So again, I want you to take a minute and think about how you would answer that question. Pause your video if you'd like. All right, so this one's a little bit more difficult. We would be looking at the total number of plastics. So you can see that we do have a column for total, but can we add this time? No, we can't add because we have different sample sizes of fish. So for this question, we need to take the average. This data is going to be opened for you on an Excel sheet. For something like looking at different species, I would want to sort the data. I think that would make it a lot easier. One way you can sort the data is by putting it in alphabetical order. That way, all the different species would be clumped together, and you could average the total microplastics for each species. So, for example, all the mangrove snapper would be in a row, and then you would be able to average the total microplastics for that species. That's how you would answer that one. And then I also want you to take a minute and see if you can think of any other questions. So this data set gives a lot of different variables, so there's definitely other questions that you could look at. Species is a variable, so maybe you're interested in a specific species. Maybe you really like fishing for, I'm trying to think of a cool fish, spotted sea trout. Maybe you want to just look at spotted sea trout and say, what type of microplastic is the most commonly found in spotted sea trout donated to our One Health Fish Monitoring Project? You can see we give you county. So that's another variable you could potentially look at. Maybe you want to look at which county has the highest fiber microplastic count, or which county has the lowest total microplastic count. That's a possibility. We give length. So maybe you want to look at larger fish versus smaller fish. You would have to define that. So you would have to say, we're categorizing small fish as, this is just an example, 0 to 250 millimeters. You could categorize those fish, have them be your small group, and then your large group be 250 millimeters and up, and kind of compare the microplastics on length. And then, of course, we give the total microplastics or the type. So many, many different things you could look at in this data set. After you come up with your research question and you answer your research question, you're going to analyze that data. So first, again, let's just go back to this first question. Which type of microplastic is most commonly found in fish from the lagoon? That nice, easy one. Just as a review, to get the answer to that, we would add up the amount of microplastics per column. When we do that, we find out that fiber has by far the most microplastics. So we do ask you to show your analysis in a figure, whether it's a table, a bar graph, a pie chart, anything like that. You can see from both of these figures pretty obviously that fiber is our most common. So as long as it's evident, you can choose any type of figure that you feel best represents your data. And again, if you feel like you need help with that, a lot of people find Excel to be the hardest part of this. I definitely found Excel hard when I was in school. So again, we can definitely set up tutoring sessions. This was a winner that used this data set. So as you can see, that was a very wonderfully creative project that got the message across. I think one thing that could have improved this project would have been including their research question at the very beginning of the clip. But it was still amazing. It was still judged high enough to become a winner. So they were looking at the fiber microplastics. And just as I talked about at the beginning of starting this data set, the bioaccumulation throughout the food chain, he kind of demonstrated that through this animation. So as the concentration of those fiber microplastics increase as you move up the food chain. Here's another winner using that data set. This was the winner from the adult category last year. This entrant looked at both the most common type of microplastic as well as what species have the most of those microplastics. So she kind of incorporated both of those things. You'll see fiber microplastic coming out of the fish, and then this is a species of jack, which ended up having the highest concentration of microplastics on the data set she worked with. So this was a very cool project as well. Now we will briefly look at another data set. We looked kind of comprehensively at the fish one. We'll go a little quicker through this one. Just practice again, coming up with some research questions. So this is our pollution mapping data. This identifies pollution sinks and hotspots by assessing the accumulation of nutrients and toxins in both sediment and water throughout the Indian River Lagoon. So for Data Jam, the data that we are giving out is the nutrient data. So looking at phosphate, nitrate, nitrite, and ammonia. So again, we're going to do kind of similar to what we just did with fish. Let's try to think of some research questions for this data set. So again, this is kind of how it'll look when you pull it up from our website. Just like the fish, this is just a little snippet. The actual data set is much, much longer. But just using this little snippet of the data set, I want you to try to think of some research questions that you could do for this data set. Go ahead and pause the webinar and take a couple minutes to try to think of some questions. All right, so just like the fish data, there's a lot of different variables included in this data set. So we have monitoring season as well as year. So this little snippet is only showing 2021, but this project has been taking data for years. So you could look at year, you could look at season. Is one season specifically always higher in a certain nutrient? You could look at water column versus pore water. So is your ammonia higher in the water column or in the pore water throughout all your seasons and years? We give the lat and the long. So if you wanted to do like northern Indian River Lagoon versus southern Indian River Lagoon, you could do something like that. And again, each one of these different nutrients is a different variable. So just to make things not super complicated, I would focus on just one of these. So maybe just phosphate or just nitrate, nitrite, just to make the analysis a little easier, especially with graphing figures and everything. So let's practice with another research question from this data set. So I really like this research question. Are phosphate levels higher in the water column or in the pore water? So to figure that out, we would, first of all, I really like simplifying the data set. If we go back to that last slide, this was a lot more overwhelming to look at, but I cut out all the nutrients besides phosphate. So it's a little bit easier to look at, not quite as overwhelming. It's easy to cut out things in Excel. So again, that's something we can teach you how to do. So we just have phosphate displayed now. We will take the average phosphate of the water column and the pore water column. So when we do that for water column, we get 0.35 milligrams per liter, whereas for pore water, we're getting significantly higher at 5.71 milligrams per liter. So a very significant difference. So again, you'll be able to display this easily visually through figures. For my example, again, I did a bar graph or a pie chart. For either of those figures, you can tell pore water obviously has the much higher phosphate concentration. Like I said, you can do any type of figure you want as long as it gets your message across. Some common things to remember when you're making figures: make sure you include a title. And then I also really like to include axis labels. So the y-axis, you can see this has a label that says phosphate concentration. We give the unit as well, milligrams per liter. And then down here, we could put an x-axis label, but it's pretty obvious what we're measuring, water column or pore water. So basically just different water sources. For this one, I decided to put data labels where you can actually see the numbers. You wouldn't have to do that because you would still be able to tell very obviously that pore water is the higher concentration. But if these were a little more even, I would definitely suggest putting the data labels. All right. So if you decide you want to sign up for Data Jam, you're going to do so by registering your team by emailing me. So my email is jschwadron@teamorca.org. That email is displayed here on the slide. You can also find that in the guidebook on our website. So you're going to just basically tell me, yes, I want to do this, and tell me if you're individual or if you're with a team. And then if you're with a team, I want the names of everyone on that team. I can get what data set you're using later on, what your research question is later on, all of that. But I would like to at least know if you're participating ASAP. This isn't binding. You could back out if you need to, but just to give us a nice estimate for our showcase, we would like to have RSVPs. So our showcase, I haven't mentioned that super in depth yet. That is an opportunity for you to look at all the artwork that was done for Data Jam. So the elementary, the middle, the high, the adult, everything will be displayed for you and other community members to look at during our showcase. It's going to be May 8th from 4:30 to 6. The location is still to be determined, but we should know that within the next couple weeks. So you're going to, if you decide you want to participate, you're going to register, then you're going to check out the different dataset info pages. Like I said a little bit earlier, you can do that at teamorca.org. You'll navigate to the Data Jam page, and then you'll navigate to datasets. You'll get a lot of good information about all those different projects, as well as some sample questions if you click on the introduction pages. You can utilize us ORCA scientists as necessary. So again, you can set up a Zoom meeting, you can come to our Center for Citizen Science, and we can help you out with this project. Everything is going to be due on April 25th, either digitally or if it's something physical, you can drop that off to our Center for Citizen Science over by the Vero Beach Airport. So again, check out our website, teamorca.org. That's where you're going to get all the resources you need. That's where you're going to get inspiration, where you can look at old projects, look at the data sets. And thank you guys so much for coming today. If you have any questions, please reach out to me. Again, my email is on this slide. Send me an email at jschwadron@teamorca.org, and I can try to help you out with any questions you may have. Thank you guys so much for watching the webinar. I hope this was very helpful. And again, let me know if you have any questions. Thank you.